Hello, everyone. I'm using Zoom to try to see whether or not this is a useful way to record and send out lectures. And the lecture that I'm going to give is actually a practice lecture that was on my PhD thesis topic, namely making bacteria work together as a team, bacterial aggregation and gene expression. So let's go ahead and get started with this particular thing. All right. So this talk is constituted in two parts. The first part has to deal with what sorts of bacterial teams we already see in nature, and second, why and how we will be making artificial teams of bacteria, which was a major feature of my PhD research. So in nature, we very rarely see bacteria in isolation, and we frequently see them forming these very complex communities. So this is an example of a community that is near, but certainly not dear to all of us. This is the sorts of bacteria that we see on human teeth. Now, each of these bacteria is assigned a specific color, or is given a specific color, through a technique known as RNA fish, where small pieces of RNA are allowed to attach or bind. Let me back up and say that again. There are colored RNA probes which will bind and fluoresce the particular pieces of RNA which are particular to individual species. And this is what allows you to get this nice pretty picture where, for instance, all of the Corynebacterium is in purple, all of the bacteria of genus Streptococcus are in green. You have capnocytophagia in red, and so on and so forth. One thing you'll notice, again, is that there are a lot of different genuses that are present here. There's this whole group of things here. But what you'll also notice is that there is some structural organization which these bacteria are taking. So, for instance, we are taking a look at what is known as a Z-stack, or an image that is made with something known as a confocal microscope that lets us get a nice three-dimensional image of some sort of bacterial slide or some item of interest. So if we're starting at sort of the center of this bacterial ball, you can kind of think of this as being a ball with a bunch of stuff inside of the ball. If we start at the center, we can actually take individual slices and come out to the periphery. And you'll notice that Corynebacterium is actually located here primarily in the center, and Streptococcus is located out here in the periphery. Similarly, in the towards the center, we have capnocytophagia here, and again towards the periphery, we have Porphyromonas out here. Within the individual sort of folios or folds of the Streptococcus, if you take a detailed view here in image G, for instance, you can see that there are a whole bunch of other bacterial genuses that are living sort of at this interface and between individual bacteria of genus Streptococcus. So there's a very complicated picture which is shown here. And this is also something which is occurring without any sort of outside human intervention. This is something that occurs naturally, as I mentioned once before, on human teeth. So if we're going to take if we're going to be able to move to the next slide here. Let's see, what happens if I clear all of my drawings? And I go back to the mouse. All right. So again, sorry folks, some technical difficulties. I'm still trying to figure this out, so please bear with me. All right. So to give just kind of a structural picture or a nice sort of cartoony picture, we have this basal layer. This would be the enamel of the tooth that would be right about here, sort of at the bottom. Let me see if I can get this in a nice red. There's the tooth, And upon that, there are these corncob structures of cornybacterium that forms a substrate or an underlying layer upon which the other bacteria can build. Now, the environment of the human mouth is extremely complex. You've got a lot of oxygen. You've got saliva, which contains enzymes in it, which are capable of breaking up certain bacteria. You also have some very complex sugars, which are not able to be processed by all of the bacteria that are on the teeth. So you will actually see this sort of spontaneous organization where at the perimeter of this structure, again, all on the base of the cornybacterium, you have bacteria that are aerobic. So, oh dear, that doesn't work very well. So aerobic or oxygen loving. These are on the outside, but their metabolism is actually creating a pocket, this anoxic pocket, where the amount of oxygen present is actually quite low. So right here, this is where you're going to start seeing anaerobic bacteria. That's where they're going to end up hanging out. And if I can shift that over here a little bit. So once again, all of the oxygen has been consumed by these bacteria on the outside, which enables the anoxic bacteria to survive, or anaerobic bacteria to survive in this relatively low oxygen environment. So as you can see, the types of species which are on the surface or this periphery are different than the ones that are present just kind of one layer below that surface. Also, the very complex set of sugars are broken down by certain bacteria out here in the periphery, and that creates a series of simple sugars, such as lactate or acetate, and those serve as a food source for these bacteria that are in this annulus or in this inner ring that would not be able to survive using the complex sugars. So essentially, by being as part of this community, these bacteria are able to survive much more effectively than they could on their own in this sort of complex environment. All right. So let's move on to the next slide, and I'll clear all of the drawings here. Bacteria are also capable of talking to each other or communicating with each other through a process known as quorum-sensing. There are other processes by which bacteria communicate, but we're going to focus on quorum-sensing for the purposes of this talk. So in the United States Congress, when it's in session, there have to be a certain number of representatives present on the floor of the Congress in order for the House to do business. You can't just have 5 representatives walk into the House chamber in the middle of the night and legislate for the entire country. There is a necessary number of representatives who must be present for it to be constituted as a legislative body. That number is known as a quorum, or a sufficient number of representatives to do business. In bacterial terms, if you have a sufficient number of bacteria present in a confined space, those bacteria will actually behave somewhat differently than when you have them in a relatively low population concentration. So in a low density state, these bacteria take on the phenotype, I'm just going to call this the red phenotype. They are producing some kind of chemical inducer, which is shown here with these black dots, which is secreted into the surrounding environment. The entire world that these bacteria see is represented by this square. And again, in this low density state, we're saying that the phenotype of the bacteria is the red phenotype. However, and if I can get my animations to work, if you have cell division that occurs and you increase the number of cells that are within that confined space, you can actually get a phenotypical change, which I'm showing here by the individual bacteria turning from red to purple. Now that can be anything from switching from a less harmful form of bacteria into a more harmful form, such as in Vibrio cholerae, for instance, where if you have a lot of them, they start producing the toxins and poisons that lead to the classic examples, or the classic symptoms, rather, of the disease of cholera. They can also literally change colors. This is Vibrio fischeri. It's a very famous example, or a very well-known example, of a bacterial shift, of a phenotypic shift, rather, that's brought about by a quorum-sensing or population density-dependent change. So ordinarily, these are free-swimming bacteria in the ocean, and if there are not enough of them present in a given flask, you don't really see anything especially special about that culture. But if you have a large number of them, as is shown here in a picture from the lab of Bonnie Bassler, you can get bioluminescence. You can get this really beautiful sort of bluish luminescent color. All right. So how does this sort of quorum sensing system work when we're taking a look at it at a sort of molecular or biological level? So one example of this is a so-called Lux-I, Lux-R quorum sensing system, where you have here an RNA polymerase, which is just hanging out. You have here a strand of DNA. So this is DNA, not RNA. This is the genetic code, which is encoding for a particular gene of interest, which I've represented just with this kind of green square here. There is a inactive monomeric form of a protein called Lux-R. It's not doing anything particularly interesting, and the RNA polymerase is not actually transcribing or making this gene. And that's a very important thing when you talk about things like when you talk about gene regulation. Again, I'm trying to explain this for a physics and not a biology class. Suffice to say, there are a lot of genes that are inside you and me right now that are not currently switched on or not currently being expressed. We're not making the proteins that those genes actually encode for. So this is an example of a gene that is not being made at this very moment. There is no RNA, and therefore there are no downstream processes that allow for this gene to be made. However, if you add something known as acylhomoserine lactone, or AHL, you will form a multimer of this Lux-R protein. You get this sort of bunch of halves coming together here, which will then bind to the DNA in a regulatory sequence or a regulatory element known as a Lux box. And that's just a little piece of DNA that basically says, okay, this is your target, you Lux-R protein. The RNA polymerase is then recruited, which is to say it is brought in by this protein multimer. You get the production of RNA of this gene of interest, and then once again downstream processes are what allow you to make the gene of interest. So because this is AHL dependent, or dependent on that concentration of acylhomoserine lactone, the bacterium that's doing this in nature is typically making a small amount of AHL. So if you have a high concentration of AHL, that is correlated with the number of bacteria that are actually present at any one time. And what that means is that if you have a high enough concentration, you get this sort of bacteria, you have a high enough concentration of AHL, and you get this change in behavior, such as the expression of this gene of interest. So I'm going to have to move. So what are some examples of things that quorum sensing systems can do in nature? So we've already met the bioluminescent Vibrio fisheri, which is actually used by the Hawaiian bobtail squid as a form of camouflage. There's a symbiotic relationship between the bobtail squid and Vibrio fisheri. So Vibrio fisheri is harvested from the ocean by this bobtail squid. It is then allowed to grow in light organs. The squid provides it with food, and in return, the bioluminescence is used by the squid as a form of camouflage. What exactly do I mean by something like that? Well, let's say that I am a shark. So, you know, I get I'm Jaws here. This is a very friendly looking shark. You know, he's got his eyes over here, and he's got his gills. Ooh, that's actually rather more sinister looking than I thought it would be. And he's got these big old pointy teeth. You know, and he is, of course, looking for lunch. And this would make a very tasty morsel. So let's say that our shark, the eye of the shark, is here, and he is looking up at the surface of the ocean here, right? So we're all kind of under the sea, just like in Little Mermaid, and this guy's focus is upwards, so he's looking something like that, all right? Now, let's say that we have this squid, and I'm not going to try to draw 10 tentacles on this. Sorry, guys. Let's say that we have this square, basically, that's moving from right to left in this image. When that square actually gets here, it's going to basically cast a shadow, and that allows the shark to basically say, aha, here's lunch. However, if this guy is actually producing his own light, if he's producing his own glare, then that shadow doesn't actually form, and the shark is out of luck. So this is a symbiotic relationship, which is a survival strategy for the Hawaiian bobtail squid. And again, it is only possible because of quorum sensing, because that Vibrio fisheri bacterium does not actually bioluminesce until there are enough of them present in one space, right? Now, there's some kind of less colorful, shall we say, things that can happen where you're talking about quorum sensing, or when you're looking at quorum sensing. So Vibrio cholerae, which is related to Vibrio fisheri, classically has two sort of modes. It has a grow and eat mode and a get out of dodge mode. So if as the Vibrio cholerae is growing inside of the gut of a human or another host, what can end up happening is, or what does end up happening, is that the density of that particular bacterial species climbs high enough that the cells basically say, all right, there are too darn many of us. We need to find some other host. We need to expand our reach. And that is where the bacteria begin producing toxins, which will irritate the lining of the human host. And that is where you get the classic symptoms of cholera, namely raging diarrhea, as the bacteria is seeking to expand itself, to expel itself from the body into the wider environment where it can find additional hosts. You also can see in things such as Pseudomonas aeruginosa, that's another pathogenic, or rather opportunistically pathogenic bacterium, where the production of something known as a biofilm can be controlled by these quorum sensing circuits. That's essentially the secretion of a whole bunch of different proteins to the outside of these bacteria. It gets extremely sticky, and that is useful for protecting the bacteria against antibiotics or other kinds of environmental insults or things that might harm or destroy those particular bacteria. I'm not going to talk about this very much, but here's just an illustration of quorum sensing in cholera. You have a low cell density state, which is what is illustrated here on the left in A, where you have the formation of biofilms and essentially okay, let's just hang out here. Whereas in a high density state, you have high concentrations of these small molecules, which deactivate these particular proteins here, which means that no signal is transmitted to LuxU, meaning that this quorum sensing circuit is actually switched off, or rather, this is an example of negative quorum sensing, where a quorum sensing signal is used to shut things down. In the low cell density state, APHA is active, and HAPR is repressed. However, in the high cell density state, HAPR is active, APHA is not activated, or rather you could say HAPR is not repressed and APHA is not activated. And what this leads to is most relevantly, the secretion of a HAP protease, which is again the irritant that causes the bowels of the unfortunate person who has this cholera inside of them to begin the classic symptoms of cholera. So I had a little bit here that I'm talking about just sort of what is a biofilm, but a biofilm in short is a microbial community. It is a large number of cells. Sometimes they're cells of the same species, sometimes they're the cells of different species that have come together for the purposes of defense and also for the purposes of utilizing resources more efficiently. They're extremely difficult to remove even by mechanical action, and once again, their formation is often regulated by bacteria undergoing a quorum sensing response. So the things that make up a biofilm are not only the bacteria themselves, but such things as polysaccharides, so multiple sugar units that are linked together in these complex polymers. You can have proteins or even the secretion of DNA to sort of hold all of these things together. You can have proteins known as fibrins or amyloids, which can be misfolded proteins or proteins that are extremely sticky. So again, often a very complex sort of stew that we have here. And I'm going to try to see this spotlight feature. So if I'm talking about fibrin biofilm, then I can kind of outline that here. And then, yeah, so again, I'm still trying to learn how to use these tools. So please bear with me. So just a few sort of discussion questions that I'd like people to think about. First, what's an example of quorum sensing? Why we might want to prevent quorum sensing. In other words, what sort of actions might we be able to take to prevent quorum sensing, and why might that be a good idea? How might we be able to prevent these sort of quorum sensing responses? And just in general, why biofilms are a problem. And then on the flip side of that, what might be some reasons why we might want to make artificial biofilms, or even better, artificial bacterial communities, or just groups of multiple different types of bacteria that are working together, sort of in emulation of what it is we see in nature. So I'm going to answer a few of these questions in the second part of the talk, which has to do with artificial bacterial assemblies. And this was a good half of my PhD. I spent years doing this, so I hope you'll find it interesting. So why would we want to do a crazy thing like assembling teams of bacteria? I've told you that teams of bacteria can make you really sick. I've told you that they're bad. I've told you that perhaps we should think about ways of breaking them up, but there are positive reasons for using bacteria as a team. One reason are things such as bioreactors. So when I say bioreactor, I mean a group of living cells engineered to synthesize a desired product or to remove pollutants. So the advantages of using living systems to do this kind of thing, rather than some sort of chemical intervention or some sort of engineering solution, which is not using a living system, you can have very good control of a molecule's symmetry. This is something known as enantioselectivity in the business of chemistry, or in the chemistry world. Suffice to say that a lot of molecules actually have mirror images of themselves. So if you think about, say, your hand, your left and your right hand, they are mirror images of each other, but you cannot superimpose one on top of the other. And oftentimes, the left-handed variety of a particular chemical, of a particular organic chemical, will behave very differently from the right-handed variety of that very same chemical. Basically, the connections are all the same, and the formula is the same, but the symmetry is different, or the enantiomer, as we say, is different. And it's actually very difficult, or historically has been very difficult, to synthesize molecules in a way that allows for the control of this molecular symmetry, or of this enantioselectivity. And I know that any organic chemists who are watching this video are going to object and say, okay, well, we are able to get pretty good enantiomeric excess, and where were you learning organic chemistry? I was learning organic chemistry back in 2008. So I know that techniques have gotten a lot better since then, but they often involve complex chemical steps, things such as directing agents, that kind of thing. So being able to do this in mild conditions, kind of everyday conditions, such as roughly body temperature and in water, is still pretty cool. And that's one advantage that living systems give us, because living systems are very good at producing only one of the two varieties, either the left-hand or the right-handed variety of molecules. Scalability. So the processes by which you will culture, say 5 milliliters of bacteria as opposed to 5,000 liters of bacteria, are pretty much analogous. You don't have to make a lot of big changes. Whereas if you have something that's being done at a 5 milliliter chemical scale and a 5,000 liter scale, there often are a lot of things that you have to take into account. It's a little bit more complicated. Now, again, I'm sure the chemical engineers watching this video are going to object to that, at which point I would just wave my hands and say, bear with me for a minute, all right? Mildness of conditions, this I will make an argument for. Again, most of biology happens at a pH of about 7, at a temperature of 37 degrees Celsius, and in water. And you don't need to add a lot of complicated chemicals or potentially dangerous chemicals in order to make what you want to make. So this reduces the amount of chemical waste which is produced. It reduces the amount of energy that you have to put in to your particular system. So mildness of conditions is more than just a buzzword. It actually is something that people are interested in. So that's great. There's a lot of work being done right now to try to get various chemical processes into living systems. And in fact, Francis Arnold at Caltech won the 2018 Nobel Prize in Chemistry for doing exactly this sort of thing. For coming up with new ways to get biology to do some really kind of wacky stuff. But bioreactors have a bit of a problem, and that is that you have to engineer the individual cells that you're trying to work with to do things that they are not at all interested in doing. So let's say that you have a very simple 3-step reaction. You're going from some chemical A to an intermediate product B, to an intermediate product C, and then finally your product is D. In order to do this, you have to have enzymatically mediated steps, meaning that you need 3 different enzymes that you are engineering into the genetic sequence of the organism that you're using. That could be a bacterium, that could be a yeast, could even be a fungus, I suppose, if you really insisted. So those 3 steps have got to be put in. But making the enzymes that mediate these steps requires energy and nutrients. And this creates something known as metabolic drag. So essentially, you are diverting resources into making things that don't necessarily benefit the organism. You can also have something, another problem, where this initial step, this A here, might actually not be something that's acted upon by the organism of interest. So that could be a sugar, for instance, that this organism does not actually know how to eat or know how to use, meaning that you need sort of preliminary steps, sort of above A here, before you can get something which is food for that bacterium and make it into something that's actually useful. Conversely, let's say that A is food and D is not food. Basically, you're asking a bacterium to starve itself, and that means that evolution is not your friend. What do I mean by that? The cell that mutates away this particular part of the circuit, that figures out a way to short circuit this process, or I shouldn't say figures out a way. If there is a mutation that takes out one of these particular steps, the cell that has that mutation is going to have an evolutionary advantage in the culture of your group of cells. And when it has that evolutionary advantage, it's going to wind up predominating in culture, and your yield of product D ends up being low. The majority of cells would end up being those that have mutated away this entire process. So that's what I mean by saying evolution is not your friend. So if you have instead a consortia-based bioreactor, or one which is based upon a community, that's a way of reducing stress and allowing processes to be optimized individually. So instead of having all three of those steps happening in one cell, I've got cell number 1 over here on the left, cell number 2 over here on the right. We just go step A to B. We have an enzyme that's mediating that process. B is able to transfer from this compartment or this cell to this completely different cell over here, which then takes this the rest of the way. One thing which is attractive about this is, let's say there's a sugar such as xylose, for instance, that we want to be in step A. We can have a bacterium that already consumes xylose and engineer it to make an intermediate that this second guy can actually use. So that's a kind of a nice thing. You can also optimize these things individually. So I can make one cell that's extremely good at converting A into B, and then one cell that's extremely good at converting B into D. And lest you think that I'm just kind of making this up, there was a study published in 2015 that I'm highlighting here, where there were 2 types of E. coli bacteria. One of these was specifically engineered to take xylose and use it to make this DHS intermediate. This DHS intermediate could then be passed from this E. coli to this E. coli. This E. coli is instead fed with glucose, so it's using a different sort of sugar. And from the DHS, you are making these feedstock simple chemicals, which MACA, PCA. What those stand for is not really important, but what is important is if you have a bacterium which is engineered to do all of these steps from the xylose to these products, the yield or the amount that you get out is relatively low. And that's what you're seeing in this P5GS column. However, if you use 2 types of bacteria, and one of them is engineered just to go from DHS to MACA and PCA, and the other is engineered just to handle the xylose, the yield of these small molecules is much, much higher. It's a difference of between 4 and 8-fold, which is worthy of being talked about. All right. Now, the problem that you can sometimes run into is if we are to have these 2 separate compartments and we have them separated, the way that B is going to transfer is not going to be in a straight line. It's going to be through Brownian motion or diffusion. You've created a transport problem. So if you can reduce that transport problem by bringing the individual cells together, this might be a way of speeding up the reaction. right? A second reason for spatial organization has to do with enabling function in hostile environments. So we're looking at an example of spatial organization that's about 10 years old. It's from the group of Rustam Ismagilov. And what they were doing is looking at this environmental pollutant pentachlorophenol, or PCP. Spongiformes chlorophénolicum is able to oxidize the PCP into harmless degradation products. However, in environments where pentachlorophenol is often found, you also find mercury 2. So this is mercury with a +2 charge. And it's usually hanging out with PCP from effluent from paper mills, that kind of thing. And this is toxic. The mercury 2 is toxic to the S-chlorophenolic. To prevent the killing of the S-chlorophenolic, the Ismagilov group surrounded the S-chlorophenolic with Ralstonia metallidurans. It's a different species of bacterium which is capable of reducing the mercury to metallic mercury, which is much, much less toxic. So these two bacteria working together are capable of dealing with this complex situation, or this complex and hostile environment, in a way that just one of them by themselves wouldn't be able to handle it. The R-metallidurans does not oxidize PCP, and the S-chlorophenolic is killed by the mercury 2. The method they used to do this was an extruder. So they essentially had a bunch of pipes, or a bunch of teeny pipes with one in the center, and they just sort of sprayed this at a target, and you wound up with this sort of structure. All right, so that's another reason why you might want to organize bacteria. Finally, controlling material in the sort of scales that you're seeing bacterial aggregates in is, I would argue, inherently interesting by itself. So in the last 30 years or so, we've actually gotten very good at controlling how material is organized on a scale from 1 nanometer to tens of microns, or tens of micrometers. And for those of you that don't sort of deal with this on an everyday basis, a nanometer is 1/billionth of a meter. A micron is 1/millionth of a meter. And the width of a human hair is about 30 microns wide, if I'm remembering that correctly. So you're dealing with things that are anything from just barely visible to the human eye to entirely invisible to the human eye. But we've gotten very, very good at making things like honeycomb structures on the nanometer scale. We can make things like these rods that are 2 microns long. We are able to make nanoparticles that are 25 nanometers plus or minus 3, with a very, very narrow size distribution. And that's even old. We've been able to do that for a long time. Conversely, if we look at the large scale assembly of materials, we can do things to tolerances of less than half a millimeter, if you're looking at the motion of, say, a fine Swiss watch, all the way up to the construction of 30 Rock, where we have buildings that are a kilometer tall. Well, let's see, maybe not a kilometer. No, the Bruges Dubai is about a kilometer tall. Anyway, very large buildings that we're able to build in a relatively precise way. But it's this area in the middle that doesn't have as many techniques developed for handling it, or how to manipulate material at this level. And that's something known as the mesoscale gap. And the bacterial aggregates that you're about to see are on the order of about 100 microns wide. We're able to go from about 20 to about 100 microns. And this is actually something that came out of my research, where you've got, again, these kind of balls of cells. Previously, there had been methods for organizing bacteria on a wide range of scales. So everything from basically inkjet printing, where you're able to put spots of E. coli in a specific manner. The work done by the Ismagulov group, which we met previously, and even some genetically controlled methods. This is actually what very much inspired our research, was a paper that came out of Spain in 2003. Where by using these things known as leucine zippers, we'll meet those in just a moment, they were able to get clusters of bacteria. So here you have a culture of bacteria where these are free floating. If you express the Jun B protein on the surface, those bacteria stick together and crash out. And if you have Phos and Jun B together, those also will crash out. So again, this was a discussion that I held when I gave this lecture before. What might be some disadvantages of the way these aggregates have been made so far? What would be a way of improving on those disadvantages? And finally, what are some of the things you might want to control when you're making bacterial assemblies? These are things I'd like you to think about as I continue this lecture. So in our research, and this was myself and Bradley Silverman of the Terrell group, we developed design rules to control aggregate size, which allows us to control the diffusion between individual cells, or into and out of the aggregates as well. Aggregate morphology, so that's for function in hostile environments. Doing this both reversibly and irreversibly. And finally, finding a method by which the cellular phenotype can be altered when aggregation has occurred. So to make our aggregates, we used two things. We used self-associating proteins and a method by which we could get those self-associating proteins onto the surface of a bacterium. The self-associating proteins we used were this pair known as SpyTag and SpyCatcher. So this is the side chain of a lysine residue, and it is binding covalently, which is to say, for those of you who are not chemists, permanently, effectively, with an aspartic acid. And you form water here, and this is an extremely stable bond. This is an amide bond. This is actually the backbone of proteins. And this is actually a very, very cool thing that was found, again, about 10 years ago, that now everybody is doing, because you can spontaneously form these covalent bonds between individual peptides or individual proteins. That's the sort of thing that we get very excited about in the biological community. You can also use something known as a leucine zipper, which is taking advantage of hydrophobic interactions. You have a iterated or semi-iterated sequence in these peptides, and they form this sort of coil-like structure. And inside of the coil, the inside portion of the coil, these are hydrophobic residues. The outside portions are hydrophilic residues. This forms what is known as a hydrophobic patch, and these tend to stick together in a manner that is reversible. You can also get ionic interactions between these sort of neighboring residues here in this sort of spiral pair. So this is a way of bringing two halves of a protein together, or in some cases, you could imagine if I have this guy attached to one bacterium and this guy attached to another bacterium, perhaps we can bring bacteria together, eh? The surface expression system we used is something called the auto-display or auto-transporter system, where you have a protein of interest that's represented by this Pac-Man over here. You have on it a leader sequence, which tells this channel protein, which is embedded in the outer membrane of a bacterium. This is the outer membrane. This is the peptidoglycan layer. This is the inner membrane of something like E. coli. You have this protein which is embedded, and up here you have a series of instructions saying, okay, kick me out of the cell. I want you to secrete me into the surrounding environment. However, because you have a translational fusion of the protein of interest with this membrane protein, When this gets kicked out of the cell, it's tethered, it's got nowhere to go, and it just kind of hangs out here on the surface of the bacteria. And that's, again, a very old technique, has an old trick that's been used for, again, years, and we were using it to display these self-associating proteins. The aggregation construct that we came up with was you have a promoter, which is a series of instructions basically saying, okay, begin transcribing and translating the genetic instructions that are immediately downstream and turn them into proteins. You have something called a ribosomal binding site. That's where the ribosome binds onto the messenger RNA and allows you to make proteins. There's this PEL-B leader sequence. It's basically, that's the signal, kick me out of the cell. There's this 6X histidine tag. That's something that is known as an epitope. That's something that can be recognized by an antibody. And by choosing the proper antibody, we can actually say, all right, how much of our protein of interest is actually present on the surface of these cells. You have X being your protein of interest. That's either one of these two leucine zippers. So let me go ahead and write that in. This is a leucine zipper. So this is a reversible interaction, and then spy tag and spy catcher, which, as we've previously said, results in a covalent interaction, which is irreversible. Irreversible. All right. Okay. The autotransporter in this case, I think this is actually a mistake. I need to correct it in my notes. But there is a type of protease in Neisseria gonorrhoeae, which allows you to stick this sort of stuff on the surface. Okay. So we should take a very quick run through here. How are these proteins being made and how is expression regulated? This is the central dogma of biology. Just in brief, we are going from DNA, which is the code by which this is, which holds basically all of the information that the cells need to make all of the proteins, to RNA. DNA is turned into RNA by a process known as transcription. And then this messenger RNA is then translated within the ribosome into the protein. And there are two kind of primary ways by which these things are regulated. Either there's some sort of a switch which is preventing transcription from taking place, which basically interrupts the RNA polymerase. And then there's also something known as translational regulation, which is preventing the messenger RNA from being turned into protein. So let's take a look at a couple examples of each of those. There's a promoter known as the araBAD promoter, where you have this series of two proteins. This is DNA. The DNA is bent into this hairpin, and when it is bent into this hairpin, an RNA polymerase is not able to access it. It's not able to bind, and it's not able to make the messenger RNA. It's stuck at the DNA level. However, if a sugar called arabinose is present, it binds here. This protein falls off, the hairpin is relieved, and then you see transcription or the turning into messenger RNA, or the creation of messenger RNA from this strand of DNA, but again, only when this protein has been removed. So just kind of as a pop question, what kind of regulation is this? I can think about that for a minute. So this is actually an example of transcriptional regulation. This is a strand of DNA. The RNA polymerase is not active, and it does not actually transcribe this gene or turn it into messenger RNA until such time as it is recruited by the LuxR protein, which brings in the RNA polymerase, and you get the transcription of this downstream gene. There's also something which allows you to regulate translation. This is where you have a protein called Lacl. This is a strand of messenger RNA. Lac-A sits down on top of the ribosomal binding site. So the ribosome is not able to get in past the Lac-I to actually make this into a protein. However, if you add lactose or its analog, something known as IPTG, the Lac-I is removed and the RNA polymerase is capable of binding to this promoter region. So aggregation, we were able to show, is dependent on construct expression. And again, this construct expression was regulated in two ways, either using transcriptional regulation, that's the araBAD promoter, or translational regulation, and that is the Lac-I promoter. So we have a series of bacteria here, which we have also genetically engineered to glow in the dark, either green or red. That allows us to keep track of where they are and keep track of what they are doing. If we add arabinose, what we're expecting is certain of these bacteria will express one half of an associating pair. That could be, say, spy tag or spy catcher. The other bacterium will express the other half of that interacting pair. These will stick together and we will wind up with an aggregate of bacteria. So we have, again, a bunch of different cells here. We add arabinose and we're able to get them to stick in a method that's observable. Now, one way that you can do this, one way that you can regulate the sizes is to have a very large excess of one type of cell as opposed to another type of cell. So for instance, if I have 5 times as many of these green guys with this sort of configuration as these guys, if the ratio here is 5:1, the resulting aggregate is actually very small, whereas at 1:1, this aggregate is larger. There's only about a 3-fold difference in sizes or volumes, but it is an effective method. This is something that I spent quite a bit of time working on, and well, I'm proud of it. A better way to regulate this is something that my colleague Bradley came up with, which is, all right, let's take a look at this ribosomal binding site, or this RBS. Is there some way that we can make the ribosome less likely to bind to this transcript or this messenger RNA? And if we modify the ribosomal binding site, we also should be able to measure how much of this 6X histidine epitope is actually showing up on the surface of the bacterium. And you wind up with a chart that looks like this. This is a technique called flow cytometry, and the amount of fluorescence is given on the x-axis here. So basically, in the direction of this arrow, that means there is more surface protein, and the direction of this arrow, that means there is less. This is a distribution because one thing that's really cool about flow cytometry is you're able to interrogate cells individually. You can look at the fluorescent signal of every single cell that's coming across a particular microfluidic channel. And so that allows you to determine an average signal. And what Bradley found is that if you have a wild type ribosomal binding site, you have expression that is corresponding to this signal, whereas if you weaken the ribosomal binding site, you end up with a signal, you end up with this signal, with much weaker. And the difference between these is roughly half an order of magnitude. So this is about a 5-fold. Oh, good Lord. That's about a 5-fold difference. I'm going to go ahead and erase that. 5-fold. With this 5-fold difference, he was able to show that you can make much smaller aggregates. So if you have a high surface protein expression level, you get large aggregates. If the protein expression level is very small, you wind up with very small aggregates. And again, showing that you can actually control this across 2 or 3 orders of magnitude. So again, some very, very nice work there. Also, looking at the morphology of these, we're able to construct core shell architecture, meaning that we have cells that are red, they're literally colored red, with the expression of a fluorescent protein. And we have SpyTag and SpyCatcher to form this core, to form basically a bunch of these red cells stuck together. If we add green cells that have the corresponding protein on the other side here, we would expect to see a core shell structure. We'd expect to see a shell of green and a core of red. But if we insert instead a control plasmid, something that does not express anything, and there are no proteins on this surface, we would not expect to see that sort of core shell structure. We'd have a bunch of cells and we have a bunch of free-floating green ones. And we showed that we can in fact do that. So going from this core, if we add the appropriate cells, you wind up with this bullseye pattern or this kind of blotch pattern. Whereas if you have just the green cells, you really don't see that. There's a little bit of non-specific stickiness, but it's certainly not as well developed as you're seeing in image A. And we're able to quantify this. I don't feel like going over that, but anyway, when the paper comes out, I suggest you read it. We're also able to show that you can break apart bacteria that are held together by these hydrophobic interactions. So if I have, let's say, bacterium A is bound to this guy, bacterium B is bound to this guy, and then I add in a whole bunch of competing stuff. A bunch of things that will compete with this interaction, and that starts to compete in there. You can actually wind up blowing these two apart. You would wind up separating these and basically causing A and B to drift off. At least that was the theory, but how was the practice? Well, if you make your aggregates and you do not add any exogenous syn-zip, in other words, you do not add any of these leucine zippers from the outside, you get something that's about 70 or 80 microns wide. It's a little bit less large than those held together with SpyTag and SpyCatcher because this is a weaker interaction than a covalent bond. You add a little bit, aggregates look a little bit smaller, but you can pretty much blow these apart if you add enough exogenous syn-zip 17 or syn-zip of this leucine zipper. And then we can show this here, that if you go from a mean aggregate size, you can control this again about threefold or so. All right. But if you try to play the same trick with SpyTag and SpyCatcher, you have a large aggregate, and a large aggregate. There is no significant difference between the sizes that are shown in all of these. Right? So finally, we were aiming to link cellular phenotype to the presence within an aggregate by engineered quorum sensing. So we're trying to get these cells to behave differently when they're in an aggregate as opposed to when they're on their own. Why would we want to do something like this? Fewer inducing steps. So in other words, you don't want to have to instruct your bacterium to first clump together and then second do the task that you have assigned it. You would much rather have it that the aggregation is right away triggering the function that you want that bacterium to have. Second, it might be possible to engineer something that allows you to prevent the escape of individual cells. Essentially, you can build in a suicide switch in these individual bacteria. Now, yes, I know that any molecular biologist who's going to see this video is going to complain that that is relatively hard to do, and I agree with you, but it is something that can be, in theory, done. And in concept, we're actually making something like an artificial tissue. We're making a bunch of different cells with a bunch of different functions, and those functions are being altered by the presence of neighboring cells. When this happens in an individual tissue or in an organ, this is something called a paracrine effect. Where, like in the pancreas, for instance, cells are secreting hormones, they're secreting chemical signals which only travel a very short distance and will alter the behavior of the cells in the immediate surroundings. And what we're doing here is actually engineering something that does that artificially. So it might be useful in future if we're looking at something like an artificial tissue. So there are three ways by which quorum sensing might be triggered. So you have all of these cells in a box, right? And quorum sensing is dependent on the density of cells, right? So one thing you can do is simply add cells to the box. You have a higher density of cells, and their phenotype changes. You also can shrink the container. You can bring the walls in. And it's actually been shown that you can take a single cell, and if you put it into a confinement which is tight enough, it will believe that it is actually in a population of a lot of cells. Basically, if the walls are close enough, the bacterium will quorum sense itself. And that's actually kind of a really, really neat, idea. Finally, we thought what you could do is to aggregate. Basically, you bring these guys very close together, and then you get that same phenotypic change as if you had either added cells or shrunk the container. To do this, we designed an experiment, as one does, and these bacteria had two plasmids in them. They had a system with they had a plasmid with aggregation, and also a plasmid that was good for quorum sensing, or a plasmid which would trigger a quorum sensing response. For those of you who don't know what plasmids are, they're little pieces of DNA that you can insert into a bacterium and basically force the bacterium to do whatever you want. Once again, this is a gross oversimplification, but just kind of go with it. All right. It's also very important to note that the bacteria were also engineered to be red. So just kind of automatically, without any quorum sensing, without anything like that, they were expressing another fluorescent protein, and this fluorescent protein colored them red. So what we're expecting is you have a bunch of red bacteria. If you add arabinose, which is what will trigger the aggregation, you see aggregation, and you also see the expression of M. wasabi. Remember, M. wasabi, which is a green fluorescent protein, will only be turned on when quorum sensing has occurred. Our hope is that we take our red bacteria, and then we see red plus green. And we see these co-localized, the signals co-localized on top of each other. If you do not add arabinose, we would just see a bunch of free-floating cells which are not quorum sensing. They are still red. They are not expressing that green fluorescent protein. And if you add the outside inducer, that acylhomoserine lactone that we met earlier, what you would be expecting is to see, again, this phenotypic shift. So if we have no arabinose and no AHL, you look at the red channel and you look at the green channel. I false colored it blue. My PhD advisor was colorblind, so we thought about this a lot in our group. And you can see a bunch of cells here, but very, very few of them are actually expressing a green signal. We measured it, it's about 2 or 3%. But if you aggregate, that's more like it. You wind up with almost everything that's red also being green. And finally, if you add the arabinose externally, you see a larger number of cells that are both green and red, as opposed to this example where you see a lot of cells that are red but not green. we can quantify this by using something known as the Manders coefficient, and that's basically saying what percentage of red pixels in each of these images are also green. So if you see a red and a green pixel on top of each other, that means that there is a cell and that it is quorum sensing. We want this number, of course, to be as high as possible. In the uninduced case, that's again about 2 or 3%. In the induced case, that's about 25%. And in the addition of an external additive, that's about 8% or so. We were actually a little bit baffled as to why this number here is lower than this number. It could just be an artifact of the fact that it is a lot easier to see an overlay of pixels when the cells are together. But this difference is, we are convinced, real. We'll see if the reviewers are convinced as well. So what we've shown is chemical induction of aggregation, control over size, shape, and function in those aggregates. And we believe that this will be useful in the future for bioreactors, and this is a primitive demonstration of an artificial tissue. So I'm not going to talk about why I became a graduate student. That's a lecture for a different day, but it involves these two gentlemen and the University of Pittsburgh, and something known as metal organic frameworks. And you can ask me all about that. I'd like to thank the members of the Terrell Research Laboratory, both in past and present. So this is the most recent photo. This is one that was sort of in the middle, and this was the oldest photo in which I am located. And you'll notice that I've got the same goofy grin. So believe it or not, I actually had a fairly good time in graduate school. All right, so these are all of the acknowledgments. And that's basically the lecture that I wanted to give. Please let me know how it went. This is my first time doing this kind of thing, and it's a big sort of surprise, sort of a big sudden surprise, having to do with COVID and the response of Anne Arundel Community College to COVID, requiring lessons online. I'm going to be teaching physics for the rest of this term, and please let me know what's gone well and what has not. All right, thank you very much. And I will go ahead and switch this off and stop the recording.